Okay, great. Welcome everyone, as I said, to the last Reclone community meeting for 2023. Today we have a number of speakers joining us. So we have Scott from Vancouver, Joseph from the Open Science Network, Joseph Shenekji from the University of Aleppo, Relles from Ateneo, the Manila University in the Philippines, who else? Felipe Buson from the Open Bioeconomy Lab here in Cambridge, UK. But that's not to say others can't jump in and just chip in and talk about their experiences. We just wanted to get a few different experiences from all over the world to see how everyone have used their OpenDNA collections. So shall I just pick on someone to start? Moy, perhaps if you're happy to start. I know it's quite late, so we'll try and let you start and we'll work from there. We want to keep this quite informal, so I guess if you have any questions, feel free to put in chat or raise your hand, and yeah, we'll go from there. So yeah, sorry. Anyway, over to you.
I'll just share. Let me open the PowerPoint slide. If this is only a very there you go. Let me check. There you go. Okay. Okay, so I hope everybody can see my screen. Okay, so hi, good day everyone. It's actually 11 p.m. here in the Philippines. So I'm on the evening shift right now. So this is our very short presentation from the Reclone hub in the Philippines. So we were able to attend several conferences and conventions for the past month, starting around the end of November until last week. The last one we had was last week to introduce Reclone and the enzymes that we got from you guys. So for our wrap up for this year, we attended last week of November. That's Aaron and I. We attended the Philippine Society of Biochemistry and Molecular Biology 50th annual convention. And we were able to present a poster introducing the Reclone hub in the Philippines. And we were able to link with several institutes in the country, like the National Institute for MEB, the Philippine Genome Center, several state universities and health institutes who are interested in getting plasmids from Reclone, um, and hopefully to also request additional ones aside from what we currently have. So we put up a Google form for the meantime for them to sign up and request specific plasmids that we have on hand. And if there are any that they would like to request, which might not be included in our website, then we will be forwarding it to the to to our to the Excel sheet where we have the reagents that we can request. We've also introduced to them the ones that we have in the collection, which we don't have yet. So in case that they will be interested in those, um, we will also be forwarding it to you. We were also able to present in a local forum in another university in the country, the University of the Philippines Institute of Biology. Aaron gave a lecture on establishing a plasmid sharing network to promote local open manufacture and research of recombinant enzymes. And as you can see here in the photo, we have the filter paper with the plasmids. So we showed it to them just so they would know what they will also receive from us in case they will also request plasmids from the Philippine hub. So we had plenty of graduate students who are interested in using in using the reagents for their thesis. And of course, we also had professors who would like to also work on these plasmids. And then the last one that we had was just last week. Aaron was able to present as a member of the National Committee on Biosafety of the Philippines. He presented in the forum on the Horizon Scan on Biotechnology Path of the Philippines about Reclone. And one important thing to note is they were able to agree on the importance of local production of enzymes. And they've included diagnostics as one of the priority areas for biotechnology research in the Philippines. So this is a new initiative of the NCBP trying to consult with local scientists as to where the country will move forward with regards to biotechnology research in the country. We were also able to use, by the way, the local findings that we've had from the policy brief that we've done October, the previous months. So the data that we got from the local interviews from the policy brief, we were able to use it to also lobby how the government works on importation of reagents, on how the government could act better against red tape, so that we would have faster access to reagents. So these were what we have been doing for the past month since November up until last week, December. And Aaron asked me to include again, we will be asking help for our ICJEB membership, although we've already raised this into the um, Department of Science and Technology, our Ministry for Science and Technology in the country. So they will be the ones who will uh, do the processing, because we were informed that it has to be a representative of the government who should request, or who should signify the interest in joining the ICGEM. And Scott Aaron also mentioned to me his request on the secretion kit and wants the gene integration kit. So he asked me to put it on the slide so I won't forget. So I'll contact, or I'll ask him to contact you again just to remind you. So that's what we have been doing. And we've already had several inquiries from other state universities as well as to the available plasmids that we have. And as an update, by the way, we were able to transform 4 of the plasmids that were sent to us, except the BSD. I think it's not with the plasmids that we have, but with transformation itself, because we were able to check there were still plasmids. So we're planning to transform it again. If not within December, hopefully early next year. And we're gonna update you if we were able to transform the BSDLF with and without M Cherry. Though we had a previous BSDLF which was requested, I think, by Keith before, and we were able to transform that BSDLF. But the one that we just received together with the other plasmids, we will try to retransform it, hopefully, if not within December, by early next year. So with the 4 others, we were able to successfully transform them. So that's it for us in the Philippines.
Oh, thanks very much, Moy. You've touched on lots of different connections already that we have. So, for example, what I probably should have mentioned earlier, Moy and Aaron were Reclone Fellows over the last quarter of 2023. So they were working on the policy reports, which was about local production of reagents in various areas of the world. So Moy and Aaron was doing the Philippines context, and I think Diego popped in the call just now. Him and Romy, they were doing looking at the local production and the policy around that within Argentina. And I may call upon you, Diego, later to give us a little update if you have a chance. A prince who wasn't able to join us today, he was also doing kind of a policy review of local production in Ghana. So these guys were our policy fellows for this year. So we're hoping to pull that report together in a polished way and kind of present that to you guys, the community. I'm gonna call upon Joseph next if you're ready, because Joseph, him and his group in kind of Syria, Middle East, they were also pulling together some details about biotechnology companies and institutes that he's recently shared with us. So we'd like to pull that into the report as well. But I think Joseph has had some good successes with the fusion DNA polymerase that we sent to you. So it'll be lovely to hear about how you've found use of that. Yeah, shall I just pass to you, Joseph, to go?
Hello, everyone. I hope you are having good times. I want to speak a little bit about my experience with Reclone plasmids. At first, I was having trouble through my PhD thesis and research regarding expression of plasmids because in Syria, in general, we don't have genetic engineering, or if we have it, it is restricted to certain facilities, not very generalized or reachable by the main public or main scientists. So it was a very uh, new experience for me on a personal level and for the university in general. Uh, so I started to ask for plasmids and try to bring plasmids from China, from India, from whichever country that can provide me. But you know that we have restrictions also. So I had few honorable attempts without any success. And in the end, after having the reclone contacted Dr. Jenny, I was able to provide the plasmids from DHL to Aleppo city and try to work upon them to transform them into bacterial strains that can uh, express PFU polymerase fusion PFU. So after attempts, uh, let's say 20 attempts maybe, we were able to produce the enzyme, and it's a very long story short, but uh, I will say this just to say about how happy I was. So after many attempts, my supervisor said, "Okay, Joseph, I respect that you are very indulgent and you are having this PhD trouble, but we can't have any more attempts. So this is the final time you will try to express your protein with these plasmids and make the transformation. If you couldn't, then I will say, I'm sorry, you can't move on. You have to change your thesis or at least your supervisor." So, so when he told me this, I did the transformation and with heat shock protocol, and then put the cells in culture for the next day. The next day, my supervisor said, "Okay, please look because I can't even look if there is a result or not. If the bacteria grew on the antibiotic." So I went and saw the results, and the bacteria practically ate the antibiotics, and the growth was very intense. So from then on, we kept going from 8 a.m. to 8 p.m. working on daily basis because I'm doing my PhD in another city in Damascus, the capital of Syria. I'm in Aleppo North. We have like a 6, sometimes 7 hours drive to go there. So I was able to produce the enzyme, and we had to check the efficiency of the enzyme. And I will try to share with you the results that came. At first, there were no results because The PFU enzyme needed to be enhanced a little bit. The concentration was not very good. It had a lot of pollution because I'm using a very frugal type of extraction by only trying to use the heat stability of the PFU in relevance to the heat instability for the other proteins in the E. coli strain I'm expressing in. So practically by just heating and taking the floating part of the solution, I am trying to extract the enzyme. So it worked. I will show you some results, but if you could wait for me just a second.
No worries, take your time. And whilst Joseph is setting that up, I think he had a request previously about protocols about using Eva Green. So if anyone else, if anyone out there uses Eva Green or has protocols to make and use that, please do send it Joseph's way.
Okay. So I will show you so this is the enzyme after I've done a PCR with the PFU fusion. This is 0.5 microliters in a master mix. This is 1 microliter, this is 2, this is 3. And you can see that the more concentration you have, the more result you have. This is the optimized result. The previous results were a bit diverse. This is the result. You can see it's very faint, and you can't see anything very strong. And you can see from this SDS page that this is the enzyme. So it needs a lot of other purification processes to have it only by itself, but it's a work in progress. And this is another result from the enzyme. You can see the band of the PCR product. This is another result, but this was very intense. This is the plasmids. This is the POBL open vent for the fusion. It has nearly 5,000 base pairs. So these are the other enzymes. So it worked with very high efficiency, and the results are very promising for next steps of the research. This is like most of what I have to say.
So I believe that's in the PTI vector backbone. Is that correct,
Yes.
Joseph?
The PTI needs some work to be done. The open vent also worked, but the POBL, the PTI, my supervisor said, okay, now we are having some exams because in the university the midterm we have it now, so we have to postpone to next year, the December of next year, because I have to publish, and I'm also a supervisor for a lab in my university, so I can't be very much traveling to Damascus to work on it. But it is in our schedule, and we are going to work on it in order to have another option for the PFU enzymes.
Oh man, that's great news to see that you can express those both those plasmids, those enzymes, and I'm sure hopefully we can hear from all the three other hubs that we have, so the Philippines, Ghana, and Argentina hubs, because they also have the open vent enzyme to work with. So it'll be good to kind of see how it expresses in different hands. Does anyone have any questions so far for Joseph, Moy, or me in general about what's been going on? Hopefully it's not jumping all over the place too much.
Just a comment. I know how you feel when you get it to work. It's just so exciting. You know, it's woohoo, yeah. So congratulations.
Thank you. And I have to acknowledge that I don't know, maybe 60 or 70 emails between Dr. Jenny and Felipe also. Each and every time I'm asking them what to do. Is this right? Is this wrong? Should I use this or this? The concentration needs some update. So I want to thank them another time for their helpful guidance and providing the plasmids. Thank you.
Probably a good time to call on Felipe, because you guys have been, as I said, as you say, have been talking amongst yourselves and optimizing and sending those plasmids to you. So Felipe, would you like to give a bit of update about what you've been doing in the lab with the collections?
Yes. So one of the things I do here at the Open Bioeconomy Lab is manage the collections that we are intending to distribute with Reclone and between the different hubs. A lot of the stuff that we've done along with distribution is to transplant a lot of the stuff that was on the molecular diagnostics toolkit to a new backbone that will hopefully express a little bit better than what we had previously. And today, like this morning, I got some results on some expression. So I'm still kind of making the gels pretty and like analyzing what expressed and what didn't. So if we can give me like an extra 15 minutes to half an hour, I think we'll get like better information. But I think there's good results and like a lot of the things have expressed, and I can show you that in a little bit.
No worries. So shall I pass off and have Scott tell us a bit more about what he's been doing before we go back to Felipe?
Sure. Yep. Okay. So first, actually, Moy, just an update on the Bacillus subtilis. I only have the Bacillus subtilis kit, not the other one. And a while back, I've told Jenny about this, and what we're doing is sending this to Jenny so that she can prep it, and then she'll be able to distribute it both to the Philippines, but everywhere else. So I'm just waiting for the DHL waybill to be able to send it to her. But it's the Bacillus subtilis one, not the other one. I don't see that I received that. Okay, so what I'm going to go into screen sharing for my presentation, and let's see, desktop, share. Okay, and let me just close off things. Zoom always covers up the whoops, hit the wrong button. Okay, can you see my screen okay? Okay,
Yep,
so we've talked about the wish list that people have been adding sequences to, and we're very, very close to getting it synthesized. So I'm just going to give an update on it. So this wish list is funded by a grant that Jenny got with the Open Bioeconomy Lab from the University of Cambridge in their EPSRC Impact Acceleration account. And that money was transferred to Drew Endy, who founded the Free Genes program. And so we're going through Drew's account, kind of. There's been a lot of back and forth about that, but I think we've got it settled and sorted out. Basically, we're synthesizing the parts that people have proposed through the Reclone network. So we call it the wish list. And what we have included in this round are 44 clonal genes, and that's a total of almost 36,000 base pairs of sequence. So that's the E. coli open enzymes components. And then there's also aligning with my interests is my new yeast protein expression toolkit. And that's 56 clonal genes at just shy of 30,000 base pairs. Most of mine are short fragments, so that's why it's more sequences, but more clones, but less base pairs. And here we change, I don't know, what's my, there we go. So I'm not going to go through and talk about all the individual ones. I'm just going to show you the list of what to get through. But what I'll comment about is that because all of these are being released under the Open MTA, we have to go through and vet them to make sure that they are off patent in the US. And so we use a website called lens.org, and that's an amazing website if you need to search for patents or things. And they also have a database of all the sequences. So if you put a patent in that has a sequence, you have to submit the sequences to the database. And so that becomes searchable. So we can take all of these sequences and search to see if the sequence was patented, if there's either an expired patent or no patent, then it gets added to the list of what can be synthesized. So if any of the people in the group here have submitted things and they don't see it on the list, it's quite possible that it's excluded because of patent issues. But these are the first round of E. coli sequences, and I won't really go through, but it's really from people around the world, and these are more of them. So I should mention that the CD is part of the E. coli protein expression toolkit syntax, and that just says where within the syntax these parts are. All of the parts here, let me see, I think, oh yes, meant to go to this slide next. So all of these parts, they have BSA1 overhangs for Golden Gate assembly using the E. coli protein expression toolkit syntax. All of the parts have been sanitized for BSA1, BBS1, BSMB1, SAP1. Those four are the most common Golden Gate assembly restriction enzymes. And so we wanted to make these parts as universal as possible so that if somebody wanted to change the restriction sites on the end from BSA1 to something else, they could do it just with PCR. The PAC1 and the SPE1 are the two enzymes that are used in the SIVA vectors, so that these can actually be transferred to different backbones. So all of the free genes, plasmids, and all of these will be cloned into POpenV3, which is an ampicillin-resistant backbone. But there are people that actually use maybe kanamycin for their, or sorry, chloramphenicol for their level 0 backbone. And so we've included the PAC1 SPE1 sites in there to facilitate that. Also, Fernan from Chile, he created the U-loop assembly, and he requested that the parts have the U-loop primers flanking them, and so we've incorporated that. Okay, so that's the list. This is the second part of the E. coli components. On the left are a whole bunch of fluorescent protein reporters that people have requested. And on the right are some of the E. coli protein expression toolkit additions that people have reported. So there's histidine tags, an 8x histidine tag to complement the 6x version. So this one has 8 histidines. There's hemagglutinin MIC tags, some silica binding tags that are supposed to be more easier to remove the protein from. There's an R5 tag already in the E. coli protein expression toolkit, which is great if you want to anchor your protein to the silica and never get it off. There are also the PBD down at the bottom. There are 12 polystyrene binding domains that you can fuse with your gene of interest. So I think, oh, and then there's 3 plasmids that I've designed that come from this paper, and it's to allow people to make operons, and it's a recursive system, so it's not just direct Golden Gate. There's a little bit of playing around, and at some stage I'll make documentation on how to do this, but you can create an operon recursively with from one or more genes in a single operon controlled by a single promoter. So there are different options there for at least 3 that I've picked from the Kiesling paper are kind of the best ones to be able to accomplish that based on their data. Okay, so now I'm going to switch over to yeast. And some of you know that I'm the creator of the Open Yeast Collection. It's available in the Free Genes repository and through the Reclone network. What I wanted to do is create a riff on Jenny's E. coli protein expression toolkit. And here's the assembly scheme or syntax for the E. coli. And we've completely adopted that for the yeast version of this. Yeast don't use ribosome binding sites. Those are just really prokaryotic elements. And so what we've done is between the B and the T1 is created a bunch of signal secretion sequences that you can fuse to your gene. So for example, Pichia pastoris, if you can coax it to secrete your heterologous protein, you save a lot on purification. So it secretes it out. All you have to do is spin down your cells and you have your protein in supernatant. There's other stuff in the supernatant, but it's much easier to purify your protein of interest from that mix than from breaking open the Pichia pastoris and having all the guts of the cell there to purify your protein from. So these are the first round. I'm also on the iGEM's engineering committee, and they've given me a budget to synthesize some other secretion signals, which I hope to be able to pass on through to the Jenny's group for distribution through the Reclone network, and possibly at Free Genes if once they open up again. Drew's trying to aim for that. I also have a couple peroximal signal sequences. These are 3 amino acids that you can tack on to the end of your protein to target it to the peroxisome of yeast. So there are some people that are interested in that. Okay, and so these, if you're familiar with the E. coli protein expression toolkit, these are the fusion partners that are in the T1, T2 position, cellulose binding domain. So all of these are taken straight from the E. coli protein expression toolkit and then recoded. This one is actually a yeast protein that people have used as kind of a carrier to get high-level expression and folding of their gene of interest. And it also helps with secretion. So I've added that in. Secretion is finicky in yeast. In Pichia, you just have to try a number of different things. You might be lucky with the first thing you try, but sometimes you have to do a bunch of other stuff. There are some fluorescent proteins, intine. These are protease cleavage sites that yeast use. The SUMO and TV cut sites are there from the E. coli protein expression toolkit, but there's also enterokinase and thrombin, and then flexible linker. I've also included these accessory proteins, and so these you would have to build an expression vector and put these into yeast. But once you have some of these, they've been shown to help secrete your heterologous protein by changing some of the things within the yeast cells. And then, of course, the E. coli protein expression toolkit, you can fuse things to the N terminus of your gene of interest or to the C terminus. And so these are the C terminus tags. And it's essentially many of the things that are in the N terminus just designed with different tags. And I think, so I want to give a real special thanks to Ian Caven, who's a member of my lab in Vancouver, the Open Science Network Society's community lab. And he's a software developer, and he's built the automated pipeline to be able to do the coding of the codon optimization of the genes, as well as adding in all of the elements, the BSA1 sites, the U-loop primers, SP PAC1, and I think that's it. Hopefully I didn't go over time, so I'm gonna, well, actually, I'll take questions if people want, and then I'll stop screen sharing just in case people want to ask any questions.
So, I mean, these are like, there's been a lot of work that both you, Scott, and Ian has put into making this selection of DNA wish lists. And I've put in chat the spreadsheet where you've been putting in all the different requests. So as I said, we're hopefully finally pressing the go button on the order. There were a few little bureaucratic issues that we still need to resolve a little bit, but hopefully we can press the button on this side of the year and get these 100 DNA parts synthesized. So if anyone has any questions about any of the E. coli or yeast parts that have been put into the wish list, hopefully we'll be posting this on the Reclone forum shortly as well. Hopefully once we've pressed the go button, so you can see how that adds to the collections that we have available. We still have a little bit of budget to add to make a final synthesis order, so do let us know. And both Scott and Ian, I believe, with all the automated pipeline that he's done, should be able to rattle through that fairly quickly. I know recently started conversations with Robson about adding potentially benzonase, and you requested also BST, which we have, or we're talking a different enzyme there. Don't know if Robson, you're here. You can comment on this.
Hello. Hi guys, listen to me?
Yes, we can hear you.
Yes.
Well, I did not expect to be talking today, but here we go. My name is Robson. I'm from Brazil, and I do have a small startup called Next Vitro Biologicals, in which we're trying to produce local manufacturing of IVD products for research purposes and molecular biology cloning. And one enzyme that I was recently talking with the community, the Reclone community, especially Jen Molly, was the benzonase, because this enzyme is actually pretty good to clean up genetic material coming from expression of these enzymes, especially enzymes that we're going to be working with molecular biology. So the benzonase, I was just wondering with her if we were able to produce that enzyme. And even though if we have an option of patent for that, how would be the production process of these? Because, how can I express benzonase inside a E. coli cell without actually killing the bacteria? So this is actually a thing I haven't dived deep in the literature to analyze.
So yeah, go through the literature, and then the other thing is also go through the patent database to look to see, because there's a wealth of information. When somebody submits a patent, they actually have to explain how to do things in a way that somebody can actually follow. And so there's a wealth of information. They leave things out just like journal articles, but there'll be information there that will allow you to figure out a path forward with producing them. I'm still waiting to hear back from Jenny. I don't do the final review on the patents. There is an active patent from New England Biolabs where they have a fusion of the benzonase with the maltose binding protein, which that would block it for synthesis for the open MTA, in my view, but I'm leaving that up to Jenny to make that call on it.
So I see. So probably also when it thinking about the benzonase, is the freedom of operation for like not only for research purposes, but like a general open MTA would be very desirable, I think, for the whole community.
Yeah.
And also maybe there's a tricky on production because it might be produced in its inactive form, and after you purify, you can actually make inactive by cleaving some peptides or something related.
Yeah, there are lots of tricks that people have used for purifying proteins that are toxic to the cell. But
Yeah.
it is a gene that's produced by a living organism, so they figured it out. I love evolution.
Indeed. And another quick questions I actually didn't have much time to look deeper as well. I have been seeing a lot of publication with engineering BST polymerases. I began to use BST polymerase from the Open Diagnosis collection here in Brazil. We have good results with that, and we were able to sell and make some partnerships with that. But actually recently I started having trouble with that because of this. It is not a hot start enzyme, and we have a lot of false positive results with that. So I don't know if you guys have looking at other BSTs options that are maybe engineered or off patent, like for the whole community. Some PhD student maybe have been developing these, and like not commercially interested on it, just on the biology side, and maybe we can find some available option for this.
Well, if you want to do the search to see if you can come up with something, then you can get it in the second round of synthesis. There's not a huge amount of room in the second round. The first round has consumed a lot of the budget. But until we actually pull the trigger and know what the final cost is, we won't know how much room there is on the second round.
I just have a final comment here, if I may. I'm actually very into as well biotransformation and biosynthesis of chemicals from Nikolai especially. So if you guys need some assistance to design or choose the best biocatalysis for this specific purpose, I will be very happy to give some assistance because it's very tricky to produce chemicals from, like from glucose or another substrates to add value. So yeah, I'm just hit me up and I can assist you guys with that as well.
Great, thank you.
Perfect. That's great to hear that you have those that kind of expertise on board, because I think one of the main reasons of this last kind of the wrap up community meeting today was more to hear from everyone else. So, you know, we called on Moise, Scott, Joseph, and Felipe just to kind of inspire you of what they've been doing. So it'll be great to hear what everyone else in the community has been doing with different parts and what they would like to add to the collections. I'm just going to post into chat also the Reclone forum if people aren't already joined into the forum. So this will be a place where you can post your questions or post any resources that you found you want to share with the community a bit more. So yeah, it's great to hear about your questions there, Robson. Felipe, do you think you're ready to tell us a little bit more about the PTIs? I mean, you don't have to share
Yes.
anything, but it'll be great to just hear the updates.
I'm sorry, I didn't prepare a lot more stuff for this.
No worries. I think this is, as I said, a bit free form.
Fresh out of the printers, so
Fresh out of the printers.
raw data. This is good.
I just prepared like a brief presentation to explain what I've been up to. So like one of the things I'm doing here in the Open Bioeconomy Lab is to try and get our collections to everyone. Because as you may know, the Free Genes project is in hiatus and we don't really know if it's going to come back and if they're going to be able to ship things to everyone. And this directly affects the collections that we manage. So a lot of the stuff that we've been doing is trying to shift these capabilities to ourselves and to our collaborators in the other Reclone hubs. So a lot of the stuff I do is focused on getting stuff from our collections in our freezers and then trying to make automation pipelines to get them in a format that's more easily shippable to everyone that's interested in them. And then some of the collections that we have managed to at least make our own inventories here and try to make them available for a few people for now are these collections over here. For now, we are still hosting the data about all of these collections in the Free Genes project website. As I mentioned in the last slide, everything here is still a work in progress. So a lot of the metadata will change places. A lot of the actual material, DNA and strains and all that is changing places. So just be aware that things might change in the future. But this is what we're trying to make available through Reclone for now. And this is what we're going to try to make available through all the Reclone hubs during the next few months. So as I said, we're still assessing the logistics and all the systems involving all this. So we're working on a new ordering system, and new ordering forms and all that. We're not that many people working on shipping these things, so it'll be interesting to see during next year how we can kind of offload a lot of this to the other Reclone hubs and how we can coordinate with everyone to make everything work and probably get DNA faster to everyone. We are aware that a lot of people are under tight deadlines, like Joseph mentioned. There's actually like super important stuff in all our lives that depend on some DNA arriving from the mail. And we're trying to make everything easier to everyone, but it takes time to put all these systems in place. We're also trying to assess the best methods for shipping DNA, if it's putting them on plates or trying to send them as glacial stocks or trying to dry them in paper, or that's still something that we're assessing. So we don't have an exact answer for that. Like everything has drawbacks and benefits. So we're talking to many different people to see like their experiences in shipping DNA and the best way to do that in the scale that we need to do for Reclone as well. We're also working on maintaining the metadata for all the collections. So as I said, like all the data for the collections right now is on Genes. So we're working on using all the platforms to be able to share those. And everything I think is mainly going to be focused on the Reclone forum. At least, if not there, we're going to post the updates about all of this when things change on the Reclone forum. So stay tuned to that. And then optimizing everything for each Reclone node. Of course, each of the nodes is not necessarily going to have all the capabilities that we have here. So things might change from node to node, but we'll try to keep a standard for the most important things, at least. So what I mentioned before, the result that kind of came today is this. I've had mixed results with expressing a lot of the stuff from the molecular diagnostics toolkit. I've been able to express the proteins that Joseph mentioned, open vent diffusion, the two polymerases, and these are some of the other ones that I've been testing. There's some other ones in the kit as well that I still need to get to, but as we said, like this is very preliminary data. I will probably need to repeat these experiments because the normalization didn't quite work very well. Like this is pre and post induced cells. So for some of the proteins, you can see like there's a lot of expression. For some of the other ones, it's not much. So there's still something we need to optimize. Like this is just one experiment. So, but at least we see a lot of expression for a lot of these proteins. So I think that's very promising. I would also post, make a post about all this in my future experiments at the Reclone forum. We already made like a general post for the PTI expression backbone there. So if the Reclone nodes or anyone else that we send things in that backbone has any experience, like good experiences, bad experiences with the backbone, please go to the forum, post things there about it, because I think the forum's going to be the main place to share information about that. That's pretty much it for me. Just a short update, but we're still working on a bunch of things. Yes. Do we have any questions? Joseph?
Yes. What kind of purification processes are you using to purify these enzymes? Is there a main way or several ways you're using?
So I haven't got to purification yet. This is just expression in bacteria and just getting the salt fraction there. But I'll probably work on the other ones that I still have left and then try to purify whole collection.
There is another question. Recently, I've been up to something called aptamer design. So, from the general concept, I think these aptamers are very simple to produce. There are some short DNA or RNA segments, and you can easily produce. The main part is the in silico design and filtering process. So, is there a chance that there are molecular diagnostics or reporters or markers that are using aptamers that you can just put inside the plasmid and produce them? I think this could be very beneficial and maybe I don't know. I don't have an experience in this, but it could be easier than enzyme production or enzyme expression. At least it can cover some aspects to be managed at the first stage of launching Reclone for the second time.
I think these could be a really good addition to the collections if they prove to be useful. I think you can maybe put them on the wish list and then we'll have to do a lot of checks to see to like improve their usefulness because if it's like a library of things then were not screened, I don't know how that will fall within our interest in like publishing it as a collection. But if they prove to be useful, all of them, we would just have to check some other things in, like to check their openness, like in terms of if they can be used for commercial purposes and like every purposes, and then put them on OpenNTA, and then we could try to synthesize them and put them together in a collection. But surely, like send us emails and post about it on the Reclone forum. I think it could be a nice proposal for the collections.
Sure, we'll do as soon as possible, hopefully.
Definitely. Thank you for the updates, Felipe. And you have covered quite a lot of things that we're going to move on to in 2024. So thank you for doing that. But as Felipe said, we do have sort of limited personnel working on the open DNA collections, at least in house. And I know there are a lot of volunteers and a lot of keen researchers out there who want to make good use of these collections. So in 2024, at the start of the year, we'll do a post on the forum just to show you what other projects are ongoing. So as Felipe said, we're trying to roll out this central ordering system so that the free hubs that we have, Ghana, Philippines, and Argentina, can help with kind of shipping them out. We're going to try and consolidate all the information we have about the collections that we have existing. So also the new set that Scott and Ian have been working on in terms of the wish list, and you guys also in the community, if you have more that you want to add to our kind of next small synthesis order. So that's other projects that we've got going on. We could always use your help with maintaining the website and the forum, just to help talk to each other and just build these connections, so that we can make best use of the collections we have already available. I know we are a bit over time, but I just want to have a last call out to check if there are any questions that you want to throw out, or any updates from anyone else in the community. And if not, I've posted in chat again our Reclone end of year survey. So should only take about 15 minutes or so. Mostly multiple choice short answer questions will be really helpful for us, for the coordination team, to figure out, what are your interests, how can we tailor what we're doing currently in the community towards projects or activities that would benefit the community members as a whole. So yeah, that's everything from my end. It's definitely nice work from Felipe. I believe that's Fernand who's chatting.
Yeah, second tomorrow from Fernand's lab.
Tomorrow, yeah. I see. Yeah. A second tomorrow. Yeah, I'm still trying to learn who else is in the community. But I don't see any more comments or questions in chat, and nothing in front of me right now. So I guess I'm just gonna wrap up and say thank you, everyone. Scott, Felipe, Joseph, Moy, all of you guys for telling us a bit about what you've been doing with the collections. For those who celebrate Christmas, I hope you have a lovely Christmas and New Year's, and we look forward to seeing you all again in 2024.
Yeah, thanks everyone. Happy holidays.
Happy holidays.
Great. Thank you very much. Happy holidays.
Thanks, everyone.
Bye.
Thank you.